Okay, hello everyone. Let us get started for today. I hope my screen is visible. Uh, just give me one sec. Yeah, okay. Welcome everyone to week 7 of our molecular biology lecture series. And today we are going to talk about translation. As you would know, this is a PMRF-NPTEL session in which we solve the previous year's run of previous year's quiz for this week and get an idea on how to solve the questions and also get an idea of the concepts behind the questions. I will switch on my earphone so that you can hear me better. I hope that is better. Okay, brief introduction about me. I'm Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur and I am taking these sessions. So as you would know, how we do this is that I show you a question, a quiz from the previous year's run of this week, and we try to solve it together. I will give you some time to figure it out and post the answer, and then we will show you the answer and then learn about the concept behind it. Okay, so the first question for today is AUG is the usual start codon which codes for which amino acid? Tryptophan, methionine, valine, or leucine. You can put whatever you think is the correct option in the chat. So you can put in whatever you think out of A, B, C, D will be the correct option. Jyoti says it is B. Anyone else? Bhavna agrees it is B. Ruksha also says it is B, which is methionine. Anyone else? Okay, so let us look at it. So brief introduction about translation. So we have already gone through two of the basic fundamentals of the central dogma of life, which is replication and transcription. Today we are going to deal with translation. Translation is the process of conversion of synthesizing a peptide using the mRNA template that was made from DNA. So how does it work? So we know that transcription is the process from which you take a gene from the DNA, you make a mRNA, is which is a copy of the information from the DNA. And using this information from the mRNA, you actually make your protein, which will do actual function. So as we have been conversing, DNA is the blueprint, mRNA is essentially the messenger in between, and protein is the final uh, worker that will actually do different processes in the cell, such as it will act as enzymes, help in metabolism, repair the cell membrane. Everything is done by proteins, but they are made from information from the DNA, which is converted to first mRNA and then to protein. So during synthesis of the protein, the main important fact that you have to keep in mind is that they are made of amino acids, which have a general structure like this: R, C double bond O, and OH. So no, not really. This is the amino acid. So all amino acids have a general basic backbone which look like this. We have amino acid group, followed by a C, double one O, and an N. This is called the peptide bond, which will then connect to the next amino acid. And essentially, piling up amino acid one of the after the other, essentially a polymerization reaction is how you make a long peptide. And we will learn how exactly the cell makes proteins using the mRNA code today. So translation can be divided, the topic can be divided into different parts. First is structure of the translation machinery. We'll learn how exactly translation works using the tRNAs and the ribosomal RNAs. We will also touch upon the genetic code, that is the codons, the mechanism of the translation, and essentially all the different nitty-gritty details of translation. So first, the structure of the translational machinery, and the most important part of it is going to be the mRNA. So we have already made the mRNA, but we will go through the general structure of the mRNA first. When we were talking about just transcription, we were just looking at how our RNA polymerase sits on the DNA template and makes a long RNA chain out of which the introns are spliced out and the exons are then joined together to form a mRNA. Now we take a closer look. The mRNA is divided into different parts. First, you would remember that eukaryotic mRNAs have a 5' guanine cap, which prevents exonuclease activity and helps in stabilization of the mRNA. It also actually helps and is very important in translation in eukaryotes. We will learn how that works. Then we have something known as the 5' UTR, or the 5' untranslated region. This region, the ribosome does not use to make a protein. The synthesis of protein only starts when the ribosome goes and recognizes a start sequence. So this is known as the start codon, and this is this can be different things, but generally it is the codon AUG. This three triplet of nucleic acids is where the initial first amino acid is added. After that, the ribosome will keep adding, essentially keep reading the mRNA sequence by traveling along the coding sequence and keep making peptides corresponding to it. And then finally, at the stop codon, it will fall off. So our ribosome starts essentially at the 5' cap. It travels all the way to the start codon, and at the start codon only, it will start synthesizing the peptide. It will keep on elongating the peptide, reading the information in the coding sequence till it comes to the stop codon, and then it will fall off. Therefore, there is obviously going to be part of the mRNA that the ribosome never sees or never translates. This is the 3' UTR, or the 3' untranslated region, because translation doesn't happen from this region. But why is this important? So in eukaryote, there are various different isoforms of different proteins, such as, for example, for a protein which is, just for the sake of saying, HOXA1. HOXA1 is a very important gene which is used to determine the developmental body plan. So it is exceptionally important in how the embryo will develop. But HOXA1 has a different isoforms or homologs, such as HOXA1, A2, A3, A4. Similarly, there are other HOX genes, such as HOX B1, B2, B2, B3, B4, C1, C1, C2, C3, C4. So essentially, all of these proteins and genes are very identical to each other. For the cell to actually regulate how exactly one particular mRNA gets translated, they cannot use the coding sequence, because the coding sequence is going to be common amongst many similarly or conserved different genes, such as HOXA1, A2, A3, and A4. What the cell uses is the 3' UTR. For a particular transcript, the 3' UTR is going to be unique to that transcript. Since this is not translated, this can be anything, and this is unique for every one of the mRNAs. So our cell actually recognizes this 3' UTR to regulate how much of this mRNA can be translated or not translated. If you want more of HOXA1, you upregulate the translational efficiency using the 3' UTR. If you do not want much of HOXA2, you downregulate using the 3' UTR. Thus, it is what the cell uses, and we also, in molecular, not molecular biology, in developmental biology, this concept is used to downregulate various genes, again looking at only the 3' UTR. You don't want to use the coding sequence because if you use the coding sequence, that is going to be common among many genes. And if you upregulate or downregulate depending on the coding sequence, you will disrupt a lot of genes at the same time and not one. So after the 3' UTR, you have the poly A tail. We have also discussed this. Poly A tail is this string of 80 to 250 uh, adenine residues that are added at the end of the mRNA. This helps in again in the protection of the mRNA from exonucleases. Exonucleases are nucleases that act at end of the mRNAs, and since you have this huge poly A tail, by the time any exonuclease actually chews off any important information, the mRNA is probably already translated and its work is done. So mRNA has the 5' end, the 5' UTR, ribosomal binding site. For prokaryotes, there is a specific ribosomal binding site which is called the Shine-Dalgarno sequence. They don't have a 5' cap. This is only eukaryote specific. And here is where the ribosome binds in case of prokaryotes. The coding sequence is this part, start to stop, and the 3' UTR. In eukaryotes, there is also the 5' guanine cap and the poly A tail. And we will see how exactly the codons work in the next slide. So, when people actually were discovering the different nitty-gritty of the of molecular biology, they figured out that you have four different nucleic acids, which is in the DNA, which is ATGC. But they had already figured out that there were 20 different amino acids corresponding to it. So they were thinking, and then they thought that yes, if there are only four nucleic acids, so each nucleic acid could not code for one particular amino acid because obviously you have four here and 20 there. If two nucleic acids at one time coded for one amino acid, again that would not work because at most that will give you 4 into 4, so 16 amino acids that can be uniquely coded by the DNA. But we have already 20 amino acids. So by trial and error, essentially people figured out it needs to be at least three different nucleic acids coming together that code for one amino acid. So they figured out the genetic code needs to be triplet in nature, which is known as a codon. So a codon is this set of three nucleotides close to three consecutive nucleotides that translate into one particular amino acid. And this you can see right here, UUU translates for phenylalanine, UCU for serine, UAU for tyrosine. So essentially, these triplet nucleic acids, which are consecutive nucleic acids read by the ribosome, code for one particular amino acid. So the genetic code is triplet, and it is known as a codon. And yeah, this is the same thing that I just said. Now, the first amino acid, the AUG, that is the start codon, that codes for methionine. Uh, yeah, so you will see right here, AUG, AUG, AUG. Yes, AUG codes for methionine. The first start codon always codes for methionine, and thus all peptides have the first amino acid that is added, it is methionine. But interestingly, the first amino acid that is added is not a normal methionine, run of the day mill methionine. It is a modified methionine known as N-formyl methionine, and it is specific. It is specific because you do not want the same methionine to start the peptide as well as be incorporated in the middle of the peptide, because then you will not know where your start codon is. So always the initiation of translation is using a N-formyl methionine, which is a modified methionine, and any methionine after that that is phased by the ribosome actually incorporates a normal methionine. So that's the one interesting key fact. So AUG is usual start codon which codes for the amino acid methionine. Any questions? Okay, moving on. The code is blank, which suggests that each codon specify only one amino acid. So the genetic code that we just discussed, you would have seen that each of the codons only translate to one amino acid. What exactly does that mean? Is it that the code is degenerate, the code is universal, the code is unambiguous, or the code is redundant? You can put whatever you think is the correct option in the chat. Jyoti says it is C, unambiguous. Anyone else? Okay, seemingly we only have one answer, so let's just look at it. So we have seen that the triplet genetic code, okay, the genetic code is triplet, so three nucleic acids code for one amino acid, and this is known as a codon. So usual start codon is AUG, which is codes for a methionine, and the code is unambiguous. Unambiguous means that only one codon will translate to one amino acid. So if you have CUU anywhere, it will always translate to leucine and nothing else. And AUU will always translate to isoleucine and nothing else. So this is what unambiguous means. So there is no confusion. One codon will only translate to one amino acid. There is no gap or a comma in the code, so there is no stopping point. Anything that the ribosome reads as it goes along the coding sequence, it will code for a amino acid. There is no comma, full stops, nothing. Next, the code is degenerate. This is an interesting fact that you will recognize by looking at the table, that even though each codon translates for one amino acid can be translated from many codons. For example, leucine has four different codons that can code for it. Threonine has four. Isoleucine has three. Valine has four. So one codon translates to one amino acid, but one amino acid can be translated from different codons. This is why the code is known to be degenerate, meaning one amino acid can be from different uh, codons translated. This means that one amino acid has more than one codon. For example, phenylalanine and is specific to two codons, UUU and UUC. Tryptophan and methionine are the two that only have one single codon. So this brings me to an interesting question. So you can think about it. Say you went on a deep sea dive and found a particular organism, and you now wanted to figure out how which this particular new bacteria, how closely is it related to our common lab bacteria E. coli. You have two options. One, you can take the entire genomic sequence of the new bacteria and compare it to E. coli, or you can take the protein sequences and compare it to the protein sequences of E. coli. Which would give you a better idea of how closely this new bacteria is related to E. coli, comparing the genomic sequences or comparing the protein sequences. Think about it for a second and let me know. Anyone wants to take a guess? So if you want to compare two organisms, what is better to compare? The genomic sequence or the protein sequence? So Jyoti says it is the genomic sequence. Any do you want to expand on that, Jyoti? Why do you think it is going to be the genomic sequence that you want to compare? It remains the order of all nucleotides in the genome. And that is not really how it works. So whenever we are transcribing for a mRNA, the mRNA always the only part that gets translated again is only the coding sequence. So you if you want to determine the order of nucleotides in the genome, like that. So I will give you what the actual answer is. So when you want to compare two distant species or two proteins or two genes from different species, we always want to compare the protein sequence because, for example, if one particular new species has CUU in its mRNA and E. coli has CUG, then on the basis of the comparison of the nucleotides, we will determine that yes, they are different. But both of them actually code for the same amino acid. So that actually tells you that resultant protein is going to be the same, even though there is point mutations or point differences between the nucleic acid sequences. So whenever you want to compare two organisms, you always want to compare the proteins because protein sequences because the protein sequences are not degenerate. The nucleic acid sequences are degenerate. So even though there is a could be a huge difference between the nucleic acid sequences, both the genomic and the mRNA, the protein could be exactly the same. So you always want to compare the proteins. Is that fine? Any questions? Okay. Next, the code is non-overlapping. So what does non-overlapping mean? So if you have the code as AUG, CUG, and whatever else after that, AUG is going to code for one particular amino acid, and this complete codon is now going to be discarded, and then you move on to the next codon. CUG is going to be the next codon coding that is going to be coding for an amino acid. It is not going to be AUG and the first last G is taken, and then the next codon is GCU. That doesn't happen. It is always in orders of three. So AUG, you are done with placing a methionine, and then you go on to CUG, then GGU, and then you so essentially only divide it into orders of three. And finally, the genetic code is universal. That is, all life forms that we have found till now actually have the same exact genetic code. So more all organisms have UUU to be phenylalanine, and the essentially that tells you that the genetic code is universal, at least for the life that we have found yet. But there are some examples. For example, UGA is a stop codon, but it codes for tryptophan in mycoplasma and mitochondria in several species. Similarly, CUG codes for leucine in general, but in yeast mitochondria, codes for threonine. But it is more or less the same for all organisms. So genetic code is also universal. So the code is blank, which suggests that each codon specifies only one amino acid. So the code is unambiguous, which suggests that each codon specifies only one amino acid. Any questions? Okay, next. The acceptor arm of a tRNA molecule ends in the conserved sequence blank at its 3' end. The options are AUG, GGC, CCA, and UGA. You can put what you think is the correct option in the chat. Hmm. Okay, I guess no one has an idea on this one, so I will give you the answer. So, what is a tRNA molecule? So one of the most other important aspects of translation is the tRNA or the transfer RNA. The transfer RNA is the machinery that actually recognizes the sequence of that is present on the mRNA, and it also carries the amino acid corresponding to that particular codon. So this is what tRNA looks like. It is a clover leaf structure. So if you have ever seen a clover leaf, Vaishnavi says she thinks it is CCA. You are actually correct. It is CCA. We'll look at it. Yes. Okay. Bhavna also says it is CCA. Perfect. Nice. Okay. So let us first look at this. So it is a clover leaf structure. If you have ever seen a clover leaf, which is a four, uh, so clover leaf has four different leaflets, and a five, three-leaf clover is considered lucky. But this here has four different arms, and they are known as the amino acid arm. This arm ends is a CCA. This arm actually carries our amino acid. There is a D arm, there is a T5C arm, and then there is the anticodon arm. The anticodon arm actually contains three different uh, anticodons corresponding to the codon present on the mRNA. For example, if the mRNA contains the codon UUU, then the anticodon arm will contain AAA so that it can base pair. And this tRNA essentially can detect that yes, the next codon to be added is UUU. And on the other hand, so on the amino acid arm, it actually carries the phenylalanine to be added. So this anticodon arm gives us the specificity of which codon the mRNA has, and this arm essentially carries the corresponding amino acid. So this tRNA is doing a lot of heavy lifting in the entire translation procedure. So transfer RNA is a clover leaf structure in two dimension and an L-shaped structure in 3D. We will look at how that looks. And it is 73 to 94 ribonucleotides in length. So this is a RNA molecule, obviously. So ribonucleotides. The tRNA consists molecule contains a 5' terminal phosphate here, right here, and an acceptor arm that ends in a CCA terminal at the 3', which we have already seen at the amino acid arm, a D loop that contains dihydrouridine, anticodon loop, and a T arm which contains T5C, which is 5 stands for pseudouridine. So it has multiple different unique nucleic acids, which are not the Generally, we have seen ATGC or AUGC. It contains some modified ribonucleotides such as pseudouridine and dihydrouridine, and we'll see why it is necessary some slides on. CCA sequence is important as it is the recognition site for the attachment of the amino acid. So this sequence actually is the site at which the amino acid actually attaches to the tRNA, and the anticodon arm is where it attaches to the mRNA. So this is what the 3D shape of the tRNA looks like. So you can see that this is generally like overall a L kind of a shape. At this end, we have where it connects to the mRNA. At this end is where it connects to the amino acid. So each tRNA is specific to us one particular amino acid. So since depending on the anticodon, it only can code for one amino acid. It only contains one amino acid that can attach to the amino acid arm. So there are 30 to 45 different tRNAs in prokaryotes and 50 different types in eukaryotes, so corresponding to the total number of amino acids as well as the total number of codons present. Obviously, you are going to be having one tRNA per codon. So you should have close to 64. Yes, you should have close to 64 tRNAs, but actually we only find 30 to 45 or around 50 in eukaryotes in the wild. And obviously this means that for a particular for a particular amino acid, there is more than one tRNA going to be present. For example, glycine has two different tRNAs, one for tRNA glycine 1 and tRNA glycine 2. So they both of them contain can carry glycine on them, but the anticodon arm is going to be slightly different in between them. There is also this fact that people have postulated something known as the codon hypothesis, which is obviously proven, which says that if we go all the way back here, if you look at the genetic code, you will see most of so for example, if we look at leucine, it has CUU, CUC, CUA, CUG. Can anyone tell me what you see that is common between all of these that code for leucine? Exactly, the CU is common. So if we look at then threonine, ACU, ACC, ACA, and ACG. Again, what is common between all of them? Yep. So essentially, the first two nucleic acids are common in all of the codons that code for one amino acid. It is only the third which is different in all of them, but all of them now code for the same thing. This gave rise to what is known as the wobble hypothesis, WOWBLE, wobble hypothesis, which says that the third, the third nucleotide actually doesn't need to be a perfect fit for the tRNA because the tRNA structure at the third nucleotide arc actually does not contain for some cases does not contain AUGC. It contains actually a different nucleotide which is called indole, 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 which can actually be paired to a lot of other nucleic acids. So, a lot of other bases. So essentially, the first two are what majorly determine what the amino acid is going to be in the codon, and the third, more if it more or less fits, then a tRNA is there to attach to it. So this is known as the wobble hypothesis, which says that the third base actually does not base pair perfectly; it wobbles a bit, but that wobble actually allows a particular the particular that one wobble and the third base actually allows multiple codons to actually code for one amino acid. What is the location of attachment site of tRNA? So the attachment site of tRNA uh, What do you mean by the attachment site of tRNA? There is a site in ribosome, which we will see as we go on. It is called the amino acyl site or the attachment site. It is present on the ribosome, not the tRNA. Is that what you were asking, Bhavna? Okay, so we will get to it in a bit. Okay, the acceptor arm of tRNA molecule ends in the conserved sequence CCA at the 3' end. The amino acid actually attaches to the amino acid arm. So the CCA at the 3' end is where the amino acid attaches, and the anticodon arm, the last three, these three nucleotides are going to base pair with the mRNA. So if you're talking about the where the amino acid attaches to the tRNA, it is going to be the 3' CCA of the amino acid arm, not the DR, the amino acid arm. Is that fine? Yeah. One other fact that you should keep in mind is that the tRNA is actually one single linear nucleotide chain. So this is one chain which is forming a secondary structure which looks like a clover leaf, but actually it is one chain. There is no two chains here. This is one that is looped around and looped around on itself. Okay. Ribosomes are nucleoprotein particles composed of rRNA and lipids, rRNA and proteins, mRNA and carbohydrates, and tRNA and nucleotides. Which of the following are ribosomes made out of? So you can put whatever you think the correct option in the chat. Bhavna says it is B, which is rRNA and proteins. Anyone else? Devadrita and Vaishnavi also say it is B, rRNA and proteins. Anyone else? Rupa and Srijoni also say it is B, rRNA and proteins. So let us go to the next machinery involved in translation, which is the ribosome. So ribosome is this huge ribonucleoprotein complex that actually is the catalytic site for protein synthesis. This is actually the machine where proteins get synthesized. So ribosomes are made out of two parts: rRNAs, which are known as ribosomal RNAs, and proteins. But interestingly, ribosomes are one of the only places where RNA is going to be the catalytic site for something to happen. So, catalytic site, essentially, we know that what are enzymes? Enzymes are these proteins that help in different biochemical processes, and they help in increasing the rate at which these processes occur. Ribosomes are made out of rRNA or ribosomal RNA and proteins, but the RNA part of ribosome is actually what does the enzymatic action. Thus ribosomes are known as ribozymes. So ribozymes, ribozyme. So they are RNA molecules that actually do enzymatic processes, not the protein part of it. So each ribosome is made out of two subunits. So there is a larger subunit and a smaller subunit. The larger subunit, a ribosome or many different larger machinery in the cell are given names based on a particular unit known as the S unit or the sedimentation rate or the Swedberg unit. So they are known as this. So in prokaryotes or mitochondria, the chloroplast have a 70S ribosome. So this number 70S determines the sedimentation rate of a particular protein or a complex, and this number is not additive in nature. It is just a number that indicates how fast it will go down a tube when you centrifuge it very fast. So the prokaryotes, the mitochondria, and the chloroplast have a ribosome which is 70S, and it is named the 70S ribosome. And it is composed of the 50S and the 30S subunit. So This number 70s is not additive, so it is not made out of 40 and 30, it is actually made of 50 and 30, which are smaller subunits. So 50 is the larger one and 30 is the smaller one. In E. coli, the 30th subunit is made out of the 16S rRNA, 21R proteins, and 50S subunit is made out of 23S rRNA, 5S rRNA, and 31 proteins. So if we look at it closely, we can already see some interesting things in the ribosome. So the smaller subunit, both in cases of prokaryotes and eukaryotes, is what binds to the mRNA first. And then upon reaching the ribosome binding site or the ribosome recognition site, which in case of the eukaryotes is going to be a 5' cap, in case of the eukaryotes is going to be a shine Dalgarno sequence. The larger subunit, the 50S subunit in case of prokaryotes, actually comes and attaches to the smaller subunit, essentially making the entire ribosome with the mRNA stuck in between it. And the ribosome has different sites, the EP and A sites, and we'll learn how exactly these work in translation in a bit. In case of eukaryotes, the eukaryotes contain a bigger ribosome, which is the 80S ribosome, and it consists of the 60S and the 40S ribosomal subunits. So the larger one is 60S, the smaller one is 40S. 60S is made out of 28S rRNA, a small 5S rRNA, 5.8S rRNA, and approximately 50 proteins. And the 40S subunit is consists of 18S rRNA and 33 riboproteins. So, so ribosome has three important sites: the A, P, and E sites. The P site is known as the peptidyl tRNA transfer binding site. The A site is known as the aminoacyl tRNA binding site, and the E site is for the deacetylated tRNA, also known as the exit site. We will learn what all these sites do. For now, we have to know that it has three important sites: the A, P, and E sites. So ribosomes are ribonucleo protein particles composed of RNA and protein. So everyone was correct in identifying this one. Okay. Okay. So moving forward, the next question is, in translation, amino-acyl tRNA synthetase is an enzyme that activates amino acids and attaches them blank. So attaches them uh, blank to the corresponding tRNA. The options are non-covalently, covalently, reversibly, or temporarily. So you can put what you think is the correct option in the chat, and then we will discuss this one. Okay. So Vaishnavi says it is A, non-covalently. Jyoti says it is B, covalently, and Devadrita and Ruksha also say it is B, covalently. Okay, so let us look at it. So we are now going into how exactly translation works. So translation, there are multiple different steps, but all of it starts with the activation of the amino acid or charging of the tRNA. So what happens is that the tRNA, when it is floating around normally in the cytoplasm, it just is this four clover leaf shape with no amino acid on this end and no mRNA attached on this end. It is just going around happily. But when it is charged, charging it means that an enzyme known as amino-acyl tRNA synthetase actually adds the amino acid to this loop of the tRNA. So this enzyme activates the amino acid by attaching covalently to the tRNA and this is when the tRNA gets charged. And this tRNA, which has the amino acid attached to its amino acid loop, is known as an amino acyl tRNA. During this process, the amino acids are attached to the tRNA with a high energy bond and are thus known as activated amino acids. So the first step is you attach the amino acid to the tRNA. Then everything else will come into the picture, but the first step is activation of the tRNA. So amino acids plus tRNA plus ATP, and the enzyme here is known as the amino acyl tRNA synthase, or not synthase, it is synthetase. Amino acyl tRNA synthetase, and it will form amino acyl tRNA, AMP, and organic PPI. So in translation, amino acyl tRNA synthetase is an enzyme that activates amino acids and attaches them blankly to this corresponding amino acids or to the corresponding to the tRNAs, and it is covalent. This brings me to a question: can anyone let me know what is difference between a synthase enzyme and a synthetase enzyme? I'll give you a moment to think about this. Anyone knows the difference between synthase and synthetase? No one has an idea what the difference between synthase and synthetase is? Anyone wants to take a guess? Okay, so a synthase is an enzyme. So a synthetase first is an enzyme that requires ATP to function. So it uses ATP to, and as a source of energy to do its job. A synthase is an enzyme that does not require ATP to function. It does its job using energy from some other source. It could be NADH or something else or a different cofactor, but it does not require ATP for its function. So since our amino acid tRNA synthetase, not synthase, uses ATP, it is a synthetase, not a synthase. Okay, next. The Shine-Dalgarno sequence blank is located 10 base pair upstream of the initiation codon. And what does it look like? There are four options A, B, C, D, and you can put whatever you think is the correct option in the chat. Bhavna says it is A, AGG, AGGU. Anyone else? Srijani also says it is A, AGG, AGGU. Okay, so let us look at it. So what is the Shine-Dalgarno sequence? So in case of prokaryotes, the Shine-Dalgarno sequence is what the smaller ribosomal subunit. Jyoti also says A. That's great. So the Shine-Dalgarno sequence is what exactly the ribosome recognizes, the smaller ribosomal subunit recognizes and attaches to onto the mRNA. So in the first step, the small ribosome subunit of the ribosome binds to the mRNA such that the initiation codon lies in the partial P site. We saw that the ribosome has a E, P, and A sites. This is done with the help of IF3, which is initiation factor 3, and Initiation factor 3 basically prevents untimely reassociation of the large and the small ribosomes, and it helps in the accuracy of initiation site selection. So, in mRNA, there is a ribosomal binding site which consists of the Shine-Dalgarno sequence and the initiation codon. Uh, this Shine-Dalgarno sequence, which is the option A, is located 10 prime base pairs upstream of the initiation codon, complementary to the region near the 3 prime end of the 16S rRNA, a component of the small subunit of the rRNA. So essentially, this is the sequence that the ribosome recognizes to bind to in case of prokaryotes. In case of eukaryotes, the sequence it does they do not have a particular sequence that it binds to, but the ribosome binds to the 5 prime cap that is present. So option A is the correct one. So the Stein-Dalgarno sequence is AGGA AGGU. So it is a very important sequence on which the ribosome actually sits. But it has not started translation yet. It is just sitting on top of it. The next question is blank is the process of mRNA encoded protein synthesis. The options are transcription, translation, replication, and splicing. You can put in whatever you think is the correct option in the chat. Bhavna says it is B, translation. Anyone else? Jyoti also says, and Srijani also says that the process of mRNA encoded protein synthesis is translation. Vivek also agrees that translation is the process of mRNA encoded protein synthesis. So eukaryotic translation, we are moving on a bit from prokaryotic translation. So translation is the process of mRNA encoded protein synthesis and is a universal process that occurs in both prokaryotes and eukaryotes. Members of both groups use information present on the mRNA, which comes from DNA by transcription, to synthesize proteins with ribosomes as the machinery. Proteins form a variety of critical functions as enzymes, structural proteins, or hormones, or therefore and are therefore crucial for biological components. Essentially, proteins are the workhorses of the cell. Everything is done by proteins. Protein biosynthesis has a key role in disease as changes and errors in the process through underlying DNA mutations or protein misfolding are key causes of underlying disease. The process by which nucleotides in the mRNA molecule direct incorporation of amino acids into a protein is known as translation. In proteins, we have already seen this. In proteins, transcription and translation are coupled. That is, translation begins while the mRNA is still being synthesized because the DNA is present in the cytoplasm directly. So once the particular gene is being transcribed, the moment the gene is being transcribed, at the same time the ribosome can sit on it on the mRNA that is being transcribed and start translating. So both of the processes are paired together in the cytoplasm. But in eukaryotes, the transcription occurs within the nucleus, and ribosomes are not present in the nucleus, they're present in the cytoplasm. And thus, when they are kicked out, the mRNA is kicked out into the cytoplasm, then only translation can start. So, ha, very nice. Translation is the process of mRNA encoded protein synthesis. Okay, next. tRNA has some unusual bases, and these are important as they protect the tRNA from blank when they're floating freely in the cytoplasm. Translation, dehydration, degradation by RNAs, or denaturation. So we have seen that tRNAs have some unusual bases such as pseudouridine or dihydroxyuridine. But why are they important? Vaishnavi and Vivek both say it is C, which is degradation by RNAs. Srijoni also says it is C, degradation by RNAs. Anyone else? Anyone else wants to take a try? Jagannath also to see degradation of RNAs. Okay, so let us look at it. So tRNA or transfer RNAs. So they combined, they are 10 to 15% of the total cellular RNA present in the cell. They're 74 to 95 ribonucleotides long and a 3.8 S sedimentation coefficient around 25,000 to 30,000 Daltons in weight. So tRNA can be have a primary structure which is linear molecule. So you have we have talked about it. It is one molecule that one long chain that gets folded in various different shapes. It has a secondary structure that looks like a clover leaf and a tertiary L-shaped 3D structure. And in addition to the usual bases AUGC, it also contains a lot of unusual bases. These unusual bases are important as they protect the tRNA molecules from degradation by RNAs when they're floating freely in the cytoplasm. Protects may and there are different other ones that protect mainly from methylation of the tRNA. These are inosine, uh, pseudouracil dihydroxyuridine. Other than the unusual bases found in tRNA, hypoxanthine, thymine, and methylguanine are also present. So essentially, they majorly prevent degradation by RNAs. Okay, moving on. The 80S ribosome dissociates to form blank subunits. 30S and 50S, 20 and 40, 40S and 60S, and 50S and 70S. So, what does the 80S ribosome dissociate to? So, 80S is the one that is present in eukaryotes. So, what does it dissociate to form? Srijani and Vivek say it is C, 40S and 60S. Okay, Bhavna and Debadrita also say it is C, 40S and 60S. Okay, so we will use this time to actually learn a bit more about translation. So we have already looked at translation initiation. So first, you charge the tRNA by attaching the amino acid to the tRNA. And second part of initiation is when the smaller subunit of the ribosome actually goes to the ribosome binding site, be it the 5' cap of the eukaryotes or the Shine-Dalgarno sequence of the prokaryotes. And now we are going to go to the elongation phase. So so translation is divided into three parts, charging four parts actually, charging of the tRNA, binding of the ribosome or initiation, elongation of the peptide, and finally termination. We are now looking at elongation. So elongation is carried out by ribosome in three different steps. First is known as decoding. Second is called the peptide bond formation and translocation. So decoding is the process in which the ribosome select and bind the incoming amino acid tRNA at a site whose anticodon is complementary to the codon of the mRNA. Decoding region is the 16S RNA. So essentially, the ribosome is scanning around along the mRNA, and when it is at a particular codon, it will wait for a tRNA to come in that will be complementary to this particular codon on the mRNA, and this process is known as decoding. The next is peptide bond formation. In this process, a peptidyl group of the P site of the tRNA is transferred to the amino acyl group in the site through the peptide bond? Okay, so first I think we look at the image. It will be easier if we look at the image. So this is how uh, elongation of a peptide occurs. First, what happens? Initially, we have seen that there are three different sites in the ribosome: the A site, the P site, and the E site. So the P site contains the elongating peptide. This is the large amino acid chain that is being formed. This is the peptide of the protein, and it is attached to a tRNA. When the mRNA ribosome moves, essentially scans and moves to the next codon, a tRNA comes in which matches the codon present on the mRNA with its anticodon loop. So if it is the codon is AGU, the tRNA is obviously carrying the amino acid that corresponds to this particular codon. Once a tRNA is has come into the A site, the ribosome, what it does is that it transfers the growing peptide chain from the P site tRNA to this particular amino acid. So this entire chain, it is attached to this tRNA at the moment. It goes and attaches to the new tRNA that is just coming to the A site. Next, the ribosome moves one codon forward. As it moves one codon forward, the P site tRNA that now does not have any amino acid attached to it goes to the E or the exit site from where it is released. The tRNA, which now has the entire peptide sitting on top of it, moves from the A site to the P site. And now the ribosome looks for a new tRNA to come in, which matches the next codon. So this keeps on going as a cycle. So again, the ribosome has a A site, which is known as the amino acyl tRNA site. Here, tRNAs which match the codon present on the mRNA come in and bind onto the ribosome. The P site, the peptidyl tRNA site, where a tRNA is present, which has the entire growing peptide chain attached to it, and the E site or the exit site, where tRNAs are released from. Once the ribosome moves to a new codon, it looks for a tRNA corresponding to that codon. That tRNA comes in. Next, the growing peptide chain on the tRNA present in the P site moves from this tRNA to this tRNA. Essentially, this entire chain transfers here. The ribosome moves one codon forward. Once it moves one codon forward, the new tRNA that has now the entire peptide chain bound to it moves to the P site, and the tRNA that does not have anything bound to it moves to the E site and is ejected via the exit site in the ribosome. So again, coming back to what is written on the slide, the decoding is the process in which the ribosome select and bind the incoming amino acid tRNA at a site whose anticodon is complementary to the codon of the mRNA, and the decoding is done with 16S RNA. Peptide bond formation. In this process, the peptidyl group of the P site of tRNA is transferred to the amino acyl group in the A site of the peptide bond. Translocation. In this case, tRNA of A site is transferred to the P site to make space for next amino acyl tRNA at the A site. The A site tRNA shifted to the E site, and this shift is coupled with the ribosome movement moving one step forward in the mRNA. So anyone has any question with how exactly elongation occurs in translation? So that was a lot of information. Take a bit of time and think about it. Anyone have any questions, please feel free to ask. Okay, it seems everyone has understood. Next, uh, after elongation, there is termination. So, how does termination occur? It occurs at the stop codon, such as UAA, UAG, or UGA. When the ribosome encounters the stop codon, there is no tRNA that is available to bind to the A site of the ribosome because there is no tRNA against the stop codon. So, once it is free, a release factor instead binds to the A site. This release factor is either RF1, release factor 1, or release factor 3. So the stop codon is recognized by release factor 1 and it binds to ribosome, and release factor 3 helps in the other termination events. Once the release factors bind, the ribosome essentially falls apart, releasing the large and the small subunits. The tRNA carrying the polypeptide is also freed up from the polypeptide product, and the ribosome is recycled, and the small subunit again goes around looking for a new mRNA molecule. And finally, we have already seen that in eukaryotes, they have a 80S ribosome, which consists of a 60S and a 40S ribosomal subunit. So 80S ribosome dissociates to form the 40S and the 60S ribosomal subunits. Any questions? Okay, moving on. For the appropriate association of a 43S pre-initiation complex with mRNA, energy has to be supplied by blank. GDP, ADP, NADH, or ATP. So 43S initiation complex is what forms in case of eukaryotes. And what does it need energy from? Vaishnavi says it is ATP or D. Vivek also says it is D. Anyone else? Devadrita also says it is D, ATP. Srijoni also says it is D, ATP. Jyoti also says it is D, ATP. Okay, so we are going to now talk about initiation of translation in case of eukaryotes. The first step that occurs is known as ribosomal dissociation. So the 80S ribosome dissociates into 40S and the 60S subunits. So this occurs so that probably occurs from a previous, the ribosome has already translated a previous mRNA, and now it needs to dissociate into two parts. And now first what will happen is that two initiation factors EIF3 and EIF1A will bind to the newly formed 40S subunit and block it being reassociating with the 60S subunit. This 40S subunit essentially is stabilized using IF3 and IF1A, a ternary complex containing the methionine tRNA. Methionine tRNA is the first methionine that is going to be attached because obviously the start codon is AUG, so first it is going to be methionine. One thing to keep in mind is that the modified N-formyl methionine is only in case of prokaryotes. In case of eukaryotes, it is just a normal methionine. And EIF2 bound to GTP attaches to 40S ribosomal subunit to form a 43S pre-initiation complex. So you have the 40S smaller subunit that binds to EIF3, EIF1A, EIF2, and the methionine tRNA to form the 43S pre-initiation complex. And EIF3 and EIF1A stabilize this complex. The binding of the mRNA to 43S pre-initiation complex results. So now the mRNA comes in and binds to the 43S pre-initiation complex to form the 48S initiation complex through the intermediate 43S initiation complex. Next, the IF4F complex is formed by the association of IF4G, IF4A, and IF4E. IF4F, referred to as cap binding protein, binds to the cap of the mRNA, IF4A and 4B bind to messenger RNA and reduce its complex structure. The mRNA is then transferred to the 43S complex, and for the appropriate association of 43S pre-initiation complex with mRNA, energy has to be supplied by ATP. The ribosomal initiation complex now scans the mRNA for the identification of appropriate initiation codon, which is the AUG. Once the 48S complex finds the AUG, the 60S ribosomal subunit now sits on top of it, sandwiching the mRNA between the two uh, 60S and 40S ribosomal subunit to form the 80S initiation complex. This binding involves the hydrolysis of GTP, which is bound to EIF2, and this step is facilitated with the involvement of EIF5, and the 80S complex is formed. The initiation factors are bound to 48S. Initiation complex are recycled and released. So essentially, there are lots of steps. First, the ribosome dissociates. Then methionine, tRNA, and other factors bind to the 40S smaller ribosomal subunit for the 43S pre-initiation complex. On the other end, EIF4A and 4B bind to the messenger RNA and bring it to this 43S initiation complex. And ATP is used to join together the 43S pre-initiation and the mRNA molecule. And then the 40S small subunit walks along the mRNA to look for the 5' AUG or the start codon. At the start codon, the 60S subunit now comes and binds to the smaller subunit and forms a 80S initiation complex. And once this complex is formed, the other initiation factors are all removed and they are recycled to go and bind to other ribosomes and mRNAs. So for the appropriate association of 43S pre-initiation complex with mRNA, energy has to be supplied by ATP. Next, the amino acyl tRNA is placed in the A site and blank are recycled to bring another amino acid tRNA. EF1A, EG1A, EF2B, or ES2U, TU. You can put what you think is the correct option in the chat. Vaishnavi says it is A, EF1A. Vivek also says it is A, EF1A. Devadrita, Bhavna, and Jyoti, as well as Ruksha, all say it is EF1A, which is what recycles and goes back and brings another amino acid tRNA to bind to the ribosomes, ribosomes A site. Can you tell me the previous question? This one again, Bhavna? Yes. So when the 43S pre-initiation complex is formed by the addition of the initiation factors as well as the methionine charged tRNA, it needs to bind to the mRNA. This mRNA binding occurs by the 43S pre-initiation complex interacting with EIF4F. EIF4F is known as the cap binding protein. So this actually occurs in two different regions. One region is where the 43S pre-initiation complex is formed. It is primed by association of elongation factors and the methionine charged tRNA. On the other side, the mRNA is bound by EIF4A and 4B. These two molecules, the proteins bind to the poly A tail of the mRNA, and EIF4F actually binds to the 5' cap. Both the ribosome and the mRNA have to come together. This actually is helped by the EIF4F complex, and they actually help in the positioning of the mRNA onto the smaller ribosomal subunit. This entire process requires energy, which is supplied by ATP. And thus, for the appropriate association of 43S pre-initiation complex with mRNA, energy has to be supplied by ATP. This is what the question asks. So, for the appropriate association of 43S pre-initiation complex with mRNA, energy has to be supplied by blank, and the answer is ATP. Any questions that you have from this? Okay, so we have a lot of A's that have been answered. So we look at eukaryotic translation in elongation. So ribosomes elongate the polypeptide chain by a sequential addition of amino acid, essentially a polymerization reaction. The amino acid sequence is determined by the order of the codons in the specific mRNA. Elongation is a cyclic process which involves certain elongation factors. So it is divided into three steps: binding of amino acid tRNA to the A site, a peptide bond formation, and then translocation. This peptide bond formation is, we will see how it works. It is very similar to how we have already seen it work in eukaryotes. So binding of uh, binding of amino acid tRNA to the A site. The ATS initiation complex contains methionine tRNA and the P site, and the A site is free. So you've already seen how the uh, even before the mRNA gets bound to the smaller ribosomal subunit, the methionine charged tRNA is already bound to the P site. Another amino acid transfer RNA is placed in the A site, so the next one. This process requires proper codon recognition of the mRNA and involvement of elongation factor 1A, which supplies GTP, and the amino acid tRNA is placed in the A site, and EF elongation factor 1A and GDP are recycled to bring another amino acid tRNA. And so essentially this is very similar to what we have already seen previously, in which we have the P site now having just the methionine charged tRNA, and the A site has the next tRNA from the next codon. The enzyme peptidyl transferase catalyzes the formation of the peptidyl bond between the methionine uh, from the P site tRNA to the next amino acid carried by the next tRNA present in the A site. This activity of this enzyme lies on 20S, 8S RNA of the 60S ribosomal subunit. It is therefore the RNA, not the protein referred to as a ribosome, that catalyzes the peptide bond formation. And the net result of the peptide bond formation is the attachment of the growing chain to the tRNA in the A site. So here you can see the tRNA in the tRNA actually contains the present in the P site contains the elongating peptide chain. Another tRNA would be here that carries the new amino acid to be attached, and the exit site is where this tRNA will go. So this amino acid chain will go to this one, and this will exit using the E site. So the amino acid tRNA is placed in the A site, and blank are replaced to bring another amino acid tRNA. So EF1A is the elongation factor which helps. I could see something moving on the screen. I have no idea what it is. Does anyone have any question? I don't think so. Okay. So the EF1A is one of the elongation factors which help in bringing new charged amino acid tRNAs to the A site. In case of eukaryotes, in case of prokaryotes, ESTU does the same thing. So this question you could have answered either way. It could be EF1A or ESTU. Okay, next. During post-translational modification, magnesium ions act as a catalyst by chelating the gamma and the beta phosphate groups, resulting in blank for phosphoryl transfer to the nucleophilic group. So what does magnesium ion do? It acts as inhibitor, deactivation, lowering the threshold of the entire reaction, or increasing the threshold of the entire reaction. Vivek says 3, lowering the threshold of the entire reaction. Anyone else? Jyoti, Devadita, and Ruksha also say it is C, lowering the threshold. So let us look at it. So now we are going to talk about post-translational modification. Vaishnavi also says C. So what are post-translational modification? So once translation has ended, which is essentially you have made the entire chain of the new protein, that is that new chain is not going to be functional as is. It is just a long chain of amino acids. To make a functional protein, you first have to do multiple things. So you have to fold the protein into a correct secondary, tertiary, and then quaternary structure. As we have seen, only the quaternary structure is the one which is functional. And also, proteins undergo something known as post-translational modification. So after translation is done, different groups on the protein or different amino acid side chains of the protein are modified. This modification is almost always essential for the functioning of the particular protein. So these could be different types: phosphorylation, glycosylation, acetylation. There are a billion things that can be done to the protein to make it functional. We are going to deal with one of the most important ones, which is phosphorylation. So what is phosphorylation? Phosphorylation is essentially addition of a phosphate group, addition of the phosphate group to a side chain of one of the amino acids of the protein. Generally, it only occurs in three different amino acids: serine, threonine, and tyrosine. And this is because they have a nucleophilic group, which is the OH group. So, if you remember your chart of amino acids, serine, threonine, and tyrosine, these only have the OH group, and the OH group is where the phosphate could be attached. So, these three are the only one with which can have phosphates. And this phosphate is donated by obviously ATP. The terminal phosphate group, which is the gamma phosphate for ATP. So if you remember your nucleotides, if this is ATP, so this is the ribose ring, this is adenosine. It has the adenosine triphosphate has three phosphate groups: alpha, beta, and gamma. The last one is known as the gamma. The gamma one is one. This is the one that contains most of the energy of the entire molecule, and this is what is transferred to the protein. So the terminal phosphate group, gamma phosphate 32 minus on the universal phosphoryl donor adenosine triphosphate, serves as the point of nucleophilic attack from the OH group, which results in the transfer of the phosphate group to the amino acid side chain. So essentially, the phosphate group gets transferred to the OH. The magnesium Mg2 plus ion acts as a catalyst by chelating the gamma and the beta phosphate groups, resulting in lowering of the threshold for phosphoryl transfer to the nucleophilic OH. Essentially, these phosphate groups are unstable due to the negative charge on it, and thus they undergo resonance. So, if you remember resonance, so this double one will go here, the minus charge will go here. So essentially, this entire three phosphates always undergo resonance amongst themselves, so they are stable. And in order to remove this group, you need a lot of energy. But if magnesium comes and binds to this O minuses present on the naked phosphates, then the energy for transfer of the gamma phosphate decreases dramatically. So it acts as a catalyst to help in transfer of this phosphate to the amino acid side chain. So during post-translational modifications, magnesium ions act as a catalyst by chelating the gamma and the beta phosphate groups, resulting in lowering the threshold of phosphoryl transfer to the nucleophilic group. Great. Next, ubiquitin is characterized by the presence of a blank and a blank. The options are N-terminal head and 5 serine residues, C-terminal tail, 7 residues, lysine residues, C-terminal tail, 5 lysine residues, N-terminal head, and 7 lysine residues. You can put what you think is the correct option in the chat. Okay, Jyoti, Vivek, and Srijoni have said B, C terminal tail, and seven lysine residues. So let us look at it. What is ubiquitin? Okay, so what is ubiquitin? Ubiquitin is a small protein which is only 76 amino acids long, and it is one it is a molecule that is also added to proteins as a post-translational modification. Majorly, so it is characterized by a C-terminal tail and seven lysine residues, and this process is known as ubiquitinization. Ubiquitinylation. It is known as ubiquitinylation. So, in this newly made protein has ubiquitin being added to its C-terminal end. But why do you need what the function does ubiquitination do? So ubiquitin is essentially the signal for the cell to actually destroy the protein it is being attached to. Essentially, proteins, if they are newly synthesized proteins, maybe there are chances that they are misfolded, which causes it to not function properly, or they have some problem in them, or they are older proteins that have been worn out and have taken damage. And such protein need to be recycled back to amino acids. The cell doesn't allow these proteins to stay in the cytoplasm and first not do their function, and secondly can do something else which is not very good for the cell. So essentially, this ubiquitin is known as also known as the garbage signal for the cell. Whenever a protein is multi-ubiquitinated, essentially multiple ubiquitin groups are added to the protein. This essentially marks the protein for degradation. So how does ubiquitination work? In ubiquitination, basically carboxylic acid of the terminal glycine from the diglycine motif in the activated ubiquitin forms an amide bond to the epsilon amine of the lysine in the modified protein and marks the cellular protein for process of degradation and via proteasome changes protein location or prevents or promotes protein-protein interaction. So polyubiquitinated protein are most certainly headed towards degradation, but monoubiquitination, ubiquitination, like one addition of one ubiquitin group, can result in the protein having change in cellular location as well. But multiple means it is going to be degraded. So ubiquitin is characterized by the presence of a C-terminal tail and 7 residue. Next, blank linkage forms between C-terminal carboxyl group of ubiquitin and E1 cysteine sulfhydryl group. What is the linkage that forms between E1 ubiquitin ligase and C-terminal carboxyl group of ubiquitin, ester thioester amide or hydrogen? You can put whatever you think is the correct option in the chat. Anyone? Vivek says it is B, thioester linkage. Anyone else? Jyoti also says it is B, thioester. Anyone else? Okay, so let us look at it. So you're still looking at how ubiquitination works. So it is a three-step process containing three enzymes, E1, E2, and E3. So first is activation of ubiquitin. which is a two-step process in which ubiquitin interacts with ATP to form ubiquitin adenylate. In the next step, it is transferred to E1 active site of the first ubiquitin cascade protein. So ubiquitin binds to ATP and then it is transferred to E1. So E1 has ubiquitin on it, and this formation causes the formation of a thioester linkage. So thioester is sulfur group attaching to one carbon and another carbon on two ends. This is a thioester. Normal esters are, obviously you would know, are one carbon on one end, a oxygen molecule, and another carbon on another end, or oxygen atom rather. Okay, so this causes formation of thioester linkage between the C-terminal carboxyl group of ubiquitin and the E1 cysteine sulfohydryl group. The next step involves transfer of the ubiquitin moiety to E2 ligase. So it transfers to E2 very easily. And then the third step is, in the last step, the ubiquitinylation cascade, there is formation of isopeptide bond between a lysine of the target protein and the C-terminal glycine of ubiquitin via one of the hundreds of E3 ubiquitin ligases. In the E1, E2, E3 cascade, one E1 molecule causes binding to several E2, which bind to E3, hundreds of E3 in hierarchical fashion. So, so why do we need a cascade like this to attach ubiquitin to a substrate? The reason is specificity. So, if we go from the opposite end, so a cell will have like thousands of proteins, which needs to be ubiquitinated so that they can head for degradation. Obviously, the same enzyme will not recognize all the proteins. So we have hundreds of different E3 ligase enzymes that recognize all these thousands of different proteins because they look for different things on all of these proteins. So 100 of E3 proteins, E3 ligases can look for which thousands of proteins need to be ubiquitinated. Now, all of these hundreds of E3 proteins are recognized by dozens of E2 proteins, which transfer the ubiquitin to them, and only there are like a handful or a couple of E1 proteins that actually attach the ubiquitin to E2. So essentially, you have a handful of E1, then dozens of E2, and then hundreds of E3, which finally attach ubiquitin to thousands of substrate proteins. So this is known as the E1, E2, E3 cascade. So a thioester linkage forms between the C-terminal carboxyl group of ubiquitin and E1 cysteine sulfohydryl group. So answer is thioester. Okay. The last question for today. Conformational changes stimulate the catalytic activity of proteins. So any protein can be activated or inactivated by blank. The options are glycosylation, methylation, acetylation, or phosphorylation. You can put what you think is the correct option in the chat. Jyoti says it is D, phosphorylation. Anyone else? Srijoni, Devadita both say D. Bhavna also says D, phosphorylation, and Vivek also agrees that it is D, phosphorylation, which allows conformation change that stimulate the catalytic activity of proteins. So we have talked about phosphorylation. It is one of the most important post-translation modification. So it is prevalent from bacteria to higher eukaryotes, sustaining in majorly two ways. First, it acts as a functional regulator of catalytic activity of proteins by defining a rigid and permanent 3D protein structure. And secondly, it temporarily phosphorylates proteins, serves as anchors for other protein substrate in signal transduction pathways. As such, it is a key player in regulation of cellular processes such as cell cycle, cell growth, apoptosis, and regulation of signal transduction pathways. Yes, so this is a lot of information. Essentially, I want to show you this. So, phosphorylation helps in two major things. First, phosphorylation helps in three-dimensional conformational changes because phosphorylation adds a negative charge to any one of the serine, threonine, or tyrosine amino acids which are originally neutral in nature. So this makes that local region of the protein hydrophilic because it is polar, and this changes how the protein will fold, and it helps in folding in the proper shape of the protein. Second, phosphorylation helps in signal transduction. Signal transduction is when a extracellular signal is given to the cell, and by transfer of phosphate groups, the signal is actually transferred on through the cell down in order for downstream cellular functions to work. For example, this is an example of the MAPK signaling pathway, in which when extracellular signal is received, initial kinase, which is inactive because it does not have any phosphate, is phosphorylated. Now, this is a active kinase. A kinase is a molecule that can donate its phosphate group. This kinase then donates it to a kinase downstream, which was inactive, and now with the addition of the phosphate becomes active. Now, this kinase will donate it to another kinase downstream, which was inactive because it didn't have a phosphate group, but active by the addition of the phosphate. And finally, this final cascade, final kinase will activate an effector by addition of the phosphate group, and then it will help in the cellular response by either growth or proliferation or gene expression or whatever else the cell needs to respond to from the external stimulus. So this is how phosphate can help in signal transduction. Any questions? Okay. So thank you for everyone for joining. This is all I had for you today. Uh, if no one has any questions, then we will end the session today here. Okay. Thank you guys. See you next week. Thank you.